We'll start out with our native lands recognition. Mononkatuk honors indigenous communities native to our chapter area, including the Pagusset, Weepawag, Quinnipiac, Tuttakat, Mononkatuk, and Hammonasset people. As we advocate for conservation of the land and its wildlife, we are indebted to the work of native and indigenous people who cherished the land for thousands of years before European colonization. And before we get started, let me just tell you that our next meeting on Zoom will be Tuesday, October 12th. Shiloh Felton, who is the field manager for Audubon's Clean Energy Initiative, will discuss how responsible offshore wind projects can protect birds in the places they live and how you can take action in your community. Now we'll get started with tonight's program. Evan Ward is head of the Department of Marine Sciences at UConn. For 30 years, he studied environmental physiology of marine suspension feeding invertebrates. Recently, his research is focused on capture, ingestion, and elimination of microplastics in nanomaterials by commercially important species and the impacts these particles have on feeding and digestive processes. So that'll be pretty much the focus of tonight's program. So Evan, welcome, and take it away.
Okay, thank you, Dennis. And I'd like to thank Carl for inviting me to give the talk today. I'm going to go ahead and share my screen now. Okay, so you should be able to see the presentation. Is that correct?
Yes, we can.
Okay, great. So yes, tonight I'm going to be talking about plastic pollution in marine environments and what I like to designate as knowns, unknowns, and uncertainties. So just a little bit of a summary of what I'm going to be talking about. First, I'm going to speak with you about macroplastic debris in the marine environment. And then I'm going to move to microplastics in the marine waters and sediments. And this is really the area of interest for my research. And we're going to talk about a little bit about formation, quantity, and distribution, and uptake and effects on marine animals. And then I'm going to walk you through one of our studies in which we're looking at microplastic interactions with bivalve shellfish, basically a case study, if you will, looking at the capture, ingestion, and egestion of microplastics by several species of bivalves. And then we'll end with a little bit of discussion regarding ingestion of microplastics by humans. So the story really begins with plastic revolution. And this is a classic scene, a picture from the classic scene from The Graduate, where Mr. McGuire is giving advice to Benjamin Braddock, who is Dustin Hoffman there, and he has one word for Dustin Hoffman, and that's plastics. And hopefully Benjamin Braddock took Mr. McGuire's advice, because if he invested heavily in plastics, he would be pretty rich today. It's really amazing to think that prior to 1950, there was very little plastic material in our lives. There was Bakelite that was patented in the early 1900s, but it was mostly used for insulators and specialty products. Since 1950, you can see what has happened with the global plastic production, as shown here. And right now we are right about here, 2021. So the estimated global plastic production is about here. That's about 450, closing in on 500 million tons of plastics that are produced annually. If we go to a little scheme showing sort of the through flow of plastic materials, it starts out fossil fuels, of course. Then it's used to create a number of different plastic materials, variety of different polymer types. It's estimated that there's been over 8.3 billion metric tons of plastics produced since 1950. Only about 9% of that has been recycled, and only about 1% has recycled more than once. Estimated 12% has been incinerated, and the rest, about 79%, has accumulated in landfills or the environment. And you can sort of see that right here. So there's a lot of plastic material that is still in the environment, either in landfills or floating around in their oceans or in the woods or other places. So if we look at the macroplastics in the ocean, and the definition here is macroplastics are greater than about 1/25 of an inch. That's approximately 1 millimeter in size. So in 2010, it was estimated that 8 million metric tons of plastic waste had entered the oceans. So again, we're about here to 2020. And so if we take a look, this is the high estimate, the mid estimate, low estimate. So if we take the mid estimate, it sits about 100 million metric tons of plastic waste are currently in the ocean. And we've all seen scenes like this from around the world, and you don't have to look too far to see some of this plastic waste on our beaches and in our marshes, etc. So another way to think of this, because 100 million metric tons is tough to conceptualize, even for me, but it's equivalent to 71 million Volkswagen Beetles in the ocean. Or another way to think of it, if for a year you dumped a truckload of plastics into the ocean every minute for a year, that would be approximately 100 million metric tons of waste. So pretty shocking. Now, there are certainly hot spots in the world where there are more plastic materials floating around than others. Some of the hot spots here around China and the Indo-Pacific here. But before and here in North America, we're not doing too badly, although we still have a lot of plastic waste in our environment. But before we start patting ourselves on the back, let's consider where a lot of this plastic material comes from. And if we look at that, we see the United States is one of the top exporters of plastic material. So basically, we are packaging our products in plastic, shipping them to countries that have very little infrastructure to deal with this plastic, and that's where it's ending up. So we're all to blame for the plastic problem in the world. So as Richard Thompson, a colleague of mine, stated in a microplastic workshop a number of years ago, plastics are not the problem. Rather, it is the way we use them that is the problem. So let's take a look at macroplastics and the impacts on marine life. Many of these pictures you've probably seen elsewhere. They are the poster children, if you will, for macroplastic debris and the effects. And it's estimated that over 800 species have been contaminated by plastics, either through entanglement, colonization of the plastic material, and or ingestion. And the impacts on megafauna are severe. Impacts on turtles and birds. Here's a bird, this is from Chris Jordan's work. An albatross here with just stomach is just packed with plastic material that is picked up, confusing the plastic material as food. If it doesn't kill the organism outright, a lot of times this bird will go to the chicks and deposit plastic material in the chicks' mouths, and then the chicks die as well. So birds, fish, all of these are well documented. So there certainly is a consensus that macroplastics are a widespread problem, and macroplastic debris affects marine animals worldwide. Let's turn our attention now to microplastics. So these are plastic particles that are less than 1/25 of an inch. And this is really an umbrella term for a variety of plastic particles. Different polymer types, different shapes, different sizes, And we can break microplastics down into two categories. The first is primary microplastics, and these are things like microbeads and personal care products, basically microplastics that were produced for a specific reason. We don't really have to worry about these as much now because in the United States and in many other countries, microbeads and personal care products have been banned. So the concentration that we see in the environment will slowly decrease over time. More importantly, we have to turn our attention to secondary microplastics. These are microplastics that form from fragmentation of larger plastic material. And this is really problematic because we see all of these large plastic materials in the environment. And over time, sunlight, UV radiation, and weathering causes those to break down into smaller particles, which then break down into even smaller particles, et cetera, et cetera. So the formation of nano and microplastics, so nanoplastics would be even smaller than microplastics, is a problem. And these materials, when they break down, have a variety of different shapes. They can be films, fibers, spherical, angular particles. A lot of these particles come from clothing, ropes, and other material. So that's really going to be the focus of the rest of the talk, microplastics. And a study that was conducted, or a paper that was published from numerous studies in The Guardian a number of years ago, has the headline, Microplastic Pollution Revealed Absolutely Everywhere. So what that is saying is anywhere anyone has looked in the world, from the deepest oceans to jungles to nearshore environments, you can find microplastics. Microplastics are pretty much everywhere. So what is the concentration of microplastics in the ocean? Well, I wish I had a good answer for you, but it really varies considerably. It varies based on location. So areas where there's a higher population size would have more microplastics than those areas that have a low population size. It also depends on random ocean processes, how the currents and tides carry the microplastics. And the other reason that it's hard to get a handle on the actual concentration is because right now there's little standardization of the methodology used to collect water or microplastics. So over here on the right, you can see a variety of different ways that you can collect microplastics, including various types. If you compare anywhere between 1 and 20 some plastic particles per gallon, if you look at natural particles, they range anywhere between hundreds of thousands of particles per gallon. So there are a lot more natural particles out there than plastic particles, and that's a good thing. But as we move forward in time, there's going to be more and more microplastics. Also, this is likely an underestimate because most studies only examine microplastics that are about 1/1000th of an inch. And there are many microplastics that are smaller than this. So this is an underestimation. Okay, what about ingestion and uptake? Well, there have been numerous documented cases of ingestion of microplastics by filter feeders, deposit feeders, various fishes, and deep sea animals. The good news, again, is that right now the number per individual or per mass is fairly low. It varies, but it's fairly low. So if you look at bivalves, the studies that were done, you can find anywhere between 0 and 9 particles per individual in a bivalve. Worms, 1 to 3 particles per gram. There's a little standardization in the literature. Fishes, anywhere between 1 and 10 particles per individuals, and dolphins, 123 to 422 particles per individual. And this is because dolphins are ingesting many of these other organisms, so they're accumulating some of the microplastics. Higher concentrations have been reported, but The quality control during extraction and isolation is suspect. What I mean by that is that when you're doing microplastic work, you have to be very careful that you are not contaminating your samples with the microplastics that are being shed from your clothing. So that has to be taken into account. And many of those studies did not analyze the microplastics properly. So the consensus here is that marine animals are exposed to ingest microplastics. The concentration in the tissues is relatively low, but certainly many, many questions remain, and many questions remain specifically with the bioaccumulation of microplastics in various organisms. We just don't have enough information right now to make any type of definitive statement. What about the effects of microplastics? Well, certainly there have been numerous lab studies which demonstrate negative effects on zooplankton, corals, worms, shellfish, and a variety of other organisms. And these impacts include feeding and digestive processes, energy reserves. There's been demonstration of inflammation, effects on cell biochemistry, and chronic toxicity. So it really ranges the effects. And this has consequences for fitness. So if you're interrupting feeding and energy reserves, this is going to affect things like how many offspring you can produce, which is a measure of fitness. So reproduction is something that could be affected and can be affected by microplastics. However, there is a caveat. There are many caveats. First of all, in these lab studies, most of the studies have used virgin plastics, not aged. So virgin material, microplastics, are really not found in the environment. Number two is that high concentrations of plastic particles have been used. Sometimes researchers use orders of magnitude higher concentrations of plastic particles than we see in the natural environment. Additionally, most studies are relatively short-term exposure rather than chronic exposure. There has also been many studies that have had poor experimental conditions in husbandry, and very few studies, as a matter of fact, there's only 2 that I know of, used the appropriate control for similar concentrations of natural material. So for example, if you feed microplastics to a bivalve and you see an effect, is that effect due to the plastic, or is it due to any type of particle that cannot be digested? So if you gave the bivalve silt, natural silt, maybe you would see the same effect as you saw with the microplastic. So not having the appropriate controls is another caveat that we need to keep in mind when we're talking about the effects of microplastics on marine animals. Additionally, there are several studies that report no effects on bivalves, shrimp, and other marine animals. So the growing consensus is that there is a mismatch between the types and quantities of microplastics in the environment and in toxicological studies. In particular, I just want to highlight again is that most lab studies use unrealistically high microplastic concentrations. At relevant concentrations, there have been few negative impacts reported, and there are many neutral impacts reported. So the impacts on organism health is still poorly understood and something we need to spend more time researching. Okay, so let's turn our attention to my lab studies, and that is shellfish, bivalve shellfish. And we all know that bivalves are ecologically important. Most of them are filter feeders, and they interact significantly with the overlying water column. So for example, an adult oyster can filter 1 to 2 gallons of water per hour. So if you think about the animal filtering for anywhere between 12 and 20 hours per day, that's a lot of water when you consider large numbers of oysters on an oyster reef. So there is a potential to accumulate microplastics and possibly see some negative impacts. Well, bivalves are also commercially important. We know that. They're consumed by animals of higher trophic levels, including humans, So the transfer of pollutants to humans is possible. So this is one of the many reasons that my lab studies the uptake and elimination of microplastics by bivalve shellfish. Okay, before we go into the meat of the research project that I'm going to talk about, let's take a look at a bivalve just so we're all sort of on the same page. This is a typical bivalve with the left shell removed, showing the underlying organs. And most bivalves, as I said before, are filter feeders. And they use the gill here to trap particles. And then using cilia and mucus, they move those particles down the gill to these collection grooves and then towards another structure called the labial palps here, which then sort the particles. Some of those particles are ingested, and some of those particles are then rejected as pseudofeces. So collection, transport, selection, and either ingestion or rejection as pseudofeces. If the particles actually go into the mouth, then it goes through the digestive system, not shown here, and eventually exits the animal as feces. So you will see many of the slides coming up will refer to pseudofeces. Remember, that's material rejected prior to ingestion. Feces is material that has gone through the digestive process. Now, we have some background information on the uptake of microplastics by bivalves because really it's not a new study. It's not a new field, I should say. Studies with microplastics and bivalves have been going on for many years. As a matter of fact, my PhD work used microplastics. We didn't call them microplastics back then. We called them polystyrene spheres or other things. But just to refer to this paper here, which I published in 1989, in which we used polystyrene microspheres to look at the uptake when we treated those spheres with different type of food compounds. So microplastics have been used for many decades in studies in the lab. So we do have some background information. And in fact, a number of years ago, I published this paper summarizing what we know about microplastics and the uptake by suspension feeders. But there's still a lot left unknown. So in particular, what we're interested in is which types of microplastics do bivalves actually ingest, and which do they actually reject, given the range of sizes, range of different polymer types, range of different shapes, etc. See if we can get to the bottom of this. Okay, so the case study I'm going to talk to you about involves uptake by this animal here. This is Middleus edulis. It's the blue mussel, the edible mussel. And this one here, probably all familiar with, Chrysostrea virginica. This is the eastern oyster. So we did two different types of studies. One was a feeding assay, where we took the microplastics, mixed them up, and we delivered them to the individual bivalves. Then we looked at which particles were, and collected which particles were being rejected in that pseudofeces. There's some pseudofeces right there, and which were egested as feces. So we collected both feces and pseudofeces and analyzed that for the microplastics that we delivered to the animals. So those were the feeding assays. The other experiments we did involved this device, video endoscopy. This is a technique I developed many moons ago. And this allows us to use basically a medical instrument to go down and look inside of a living whole animal and look at how it is processing the microplastics. So here's a close-up of the optical insertion tube right there inside of an oyster and inside of a mussel. Now the light associated with the end of the endoscope is cold light. You can see the fiber optic cable right here running off screen. And so all of the heat associated with the light is off on the light box. So this is cold light being delivered to the animal. So that's basically the setup. All right, let's take a look at some of the results. So I'm going to show you some video from the endoscopy experiments, and this is going to show you capture and transport of plastic particles. All the video is in real time, and you're going to see a muscle. The muscle has a flat gill with one type of filament. There's the filaments there. This is the gill of the muscle is seen through the endoscope. And you can see it really does look like a filter, doesn't it? So that's the muscle gill. And then you're going to see the oyster gill. The oyster gill has this lovely plicated gill. It's thrown into folds here. The filaments are a little smaller, a little thinner, but you can still see the individual filaments there. They also have another filament type down here between the grooves, right down in here between the plica. And so they have two different types of filaments. So muscle flat and the oysters are folded or plicated. All right, let's take a look at the first video. Here we are on the outside of a muscle, and I'm delivering it some plastic particles. You can see them being drawn in. Now we're zooming in now with the endoscope into the gills of the muscle, and eventually we come up and we're going to deliver it some polystyrene plastic particles, and you can see them being captured and transported down to this groove here. Here are some pictures using larger plastic particles. You can see them also being captured. They move a little slower than the smaller particles, but the animal has no problem capturing these particles and transporting them. Here are some pictures showing fibers being captured and transported, again being transported down to this groove. Here are some smaller fibers we gave it. They're being accumulated here in this groove. These are going to go off to the labial palps. Here's the oyster. Again, different size particles. You can see no problem capturing, transporting them to this groove here, which is going to take them off to the labial palps towards the mouth. This is a dorsal groove here, so only small particles end up in the dorsal groove. Here back at the ventral groove, here are long fibers we gave it. Again, no problem capturing, transporting these fibers. And there's a large fiber and a small fiber there. So this is all microscopic. I forgot to mention that this is about 350 micrometers across here. So these particles are microscopic. You couldn't see these with your naked eye. Okay, so from these studies, we know that both species can capture and transport the microplastics. And I didn't really spend much time, but also oysters can select plastics on the gill. Mussels select only on the labial palps. Okay, next I'm going to show you the rejection of plastic particles. So we're going to be zooming in at this spot on the mussel. This is where the pseudofeces comes from. And this spot on the oyster, this is where the pseudofeces or the rejected particles come from. Whoop, sorry. And we'll roll the video. So watch carefully. This happens very quickly. So here we are, mussel first. And there's two plastic particles being rejected, larger ones. Here's two real small, these are like 20 micrometer particles being rejected. Here's a bolus of fibers, microfibers, that has been wrapped up in mucus by the mussel, and they're going to be rejected. So we see a lot of microplastics being rejected by the bivalves. This is an oyster. Oyster has a little different strategy. Instead of continually rejecting particles, it basically accumulates them into this bolus, and then periodically it will adduct its valves, clasps its valves together, and eject that pseudofeces back into the environment. So from these studies, we know that the rejection of the microplastics occurs within minutes of exposure, and so not all plastic particles that are delivered to the bivalves are ingested. They're captured, but not ingested. So we can quantify this by looking at our feeding assays. Here is the percent of particles rejected by the animals in pseudofeces. Here is the diameter of the particles that we expose them to. And we can see for both oysters and mussels, as we increase the diameter of particles, a higher percent of those particles are rejected in pseudofeces. As a matter of fact, up here at 1,000 micrometers, which is 1 millimeter, all the particles were rejected. So neither the oyster nor the mussel ingested any 1 millimeter plastic particle. They were all rejected. For fibers, a little different story. There's really no relationship between the percent of the fibers rejected and the size of, or the, I should say, the length of the fibers. All of these fibers were 30 micrometers in diameter, and that's for oysters and mussels. However, you can still see for oysters, they were rejecting about 60% of all the microfibers, and mussels were rejecting about 25% of all the microfibers. So still a large percentage of the plastic fibers were being rejected by the animals. If we look at the egestion of microplastics, those microplastics that were in fact ingested in the feces in less than 3 hours, that's this here. And the reason that's important is because if the material ingested is egested in feces in less than 3 hours, that means it does not undergo the full digestive process. It's too complicated right now to get into why that is, but if it comes out less than 3 hours in the feces, it basically took a fast track through the animal. So what we see is that for oysters and mussels, again, as we increase the size of the microplastics, those microplastics that happen to be ingested, those microplastics, a higher percentage of them are egested in feces as we increase the size, so egested in less than 3 hours. So these are really taking the fast track through the animal, and so are these. These are taking a little slower. And the microfibers, same idea here. A lot of those microfibers take a fast track through the animal. You can see that here, about 70 or so percent there, and about 55% there, except for the longest microfiber with mussels. So we're thinking that these might be getting caught in the digestive tract of the mussel. And we're following up with some additional studies on that. And those data really back up things we've seen before. So this is the ingestion of spheres by oysters and mussels in hours. Okay, so if you give them various types of microspheres and then collect their feces over time, you see that there is a rapid elimination of the microplastics in feces. Here we did the same thing with microplastics and nanoplastics. You can see again, pretty rapid decrease in the number of microplastics and nanoplastics in the feces. So after even 1 day, we're seeing a large decrease in the number in the animal. So elimination pretty quickly. So the perspective that we can get from this is that the movement of plastic particles into and out of mussels is pretty rapid. So if you consider that these are the microplastics in the environment, a mixture of spheres, fibers, fragments, and other things, and we know that the animals are being exposed to these, and we know that these particles can be captured by the gill. However, we also know that within minutes, a large portion of these, depending on their size, are rejected back into the environment. Of those that are ingested, those are egested within several hours. A large portion are egested in feces in a few hours. So consequently, if you look at a bivalve, you just go out into the environment and pick a bivalve and measure the number of microplastics in its gut. There's relatively few for these reasons: rejection and rapid elimination in feces. So because of the selective feeding capabilities of bivalves, few of those microplastics remain in their gut. Okay, so now let's turn our attention and just talk a little bit about human ingestion of plastics. I showed you a different form of this before, but we know the world plastic production continues to increase. And like other animals, humans are eating plastic particles. Um, the plastic is contaminating our food, and as there's more and more plastic being used in food processing, etc., we're being exposed to more plastic material. If you look on the web, you can see people saying that everyone eats about a credit card's worth of plastic every 2 weeks. I'm not so sure of that, but it's a good sort of headline. I'm not sure it's that much, but certainly we are ingesting plastic material without a doubt. So there's a number of pathways that plastics can get to humans. So here we are down here. Here's our food. So you can see some of the ways that plastic ends up in our food. Uptake from the environment, showed you some of that just earlier. Settling during preparation and mealtime. There are a lot of microfibers that are floating around in your house. Some of those settle onto your meals. Contamination during transport, during processing, and leaching from plastic packaging. That's a big route right there. So a variety of different ways that plastics can get into our food and then into us. There's been a recent study, relatively recent study, and that provides some data and some perspective. That's this one here by Cox. So if you look at the number of microplastics consumed, and unfortunately there's no data for red meat, poultry, dairy, grains, fruits, and vegetables, so that's a big hole in our database. But if you look at other types of food that we eat, and you look at the different, a male adult, female, male child, female child, etc., you can see that anywhere between about 40 and 50,000 microplastic particles are consumed annually. That's about a half a pound of microplastic per year. However, it's estimated that we inhale the same amount. So ingesting about a half a pound and inhaling about a half a pound of microplastics per year. If we break that down a little bit more by category, here's just a few of those categories. Remember, we don't have any data for red meat and vegetables and things like that, but there's good data for honey, salt, alcohol, tap water, sugar, bottled water, seafood, and air. You sort of get this view. So air, that's the inhalation. Seafood, remember seafood, this is not just bivalves, this is also fish. Most of that is probably contamination during processing. An interesting one is bottled water. Bottled water contains on average 22 times more microplastics than tap water. So another good reason to stop drinking bottled water. Not only are you ingesting more microplastics, but you're also contributing to the plastic problem. And the values for females is about the same. It's a tiny bit lower than the adult male. So not a lot of data, but some out there, and there's a number of studies that are ongoing right now that will provide more data to us in the near future. So some of the challenges for the future I put here. So certainly, what are we going to do? First of all, plastics, we're not going to get rid of plastics in our daily life. Plastics have made our cars more fuel efficient. They have kept our food safer in many ways. They are used in the medical industry. And so plastics are really an important part of our day-to-day business. However, what we need to do is think of ways that we can reduce the amount of plastic material, unnecessary plastic material that we use. So there's some engineering solutions. So things like plant-based polymers and green chemistry designed to degrade, to be used and then degrade after weeks or months in the environment. Also, advances in the recycling and chemical degradation of plastics is important. So instead of just getting them into a landfill, getting them into some type of a process which breaks the polymers so that the base material can be reused. Certainly there are policy and regulatory solutions. We've seen that here in the state of Connecticut. Bans on certain types of plastic materials like bags, deposits. There's a plastic bottle deposit that's going into effect, and making manufacturers more accountable for the plastic waste. And then, of course, there are social and behavioral solutions as well, education and reduce, recycle, reuse. Okay, so I'd like to leave plenty of time for questions. So a few take-home points. Certainly macroplastic loads in the oceans are increasing rapidly. That's without a doubt. And fragmentation of plastic debris produces microplastics. In the US right now, the concentrations in our waters is still relatively low, but it will be increasing in the future. Marine animals are ingesting microplastics, and some harmful effects have been documented, but we certainly need a lot more data, especially using environmentally relevant concentrations of microplastics and longer-term studies, chronic exposures. Bivalves have some protection for microplastics, and this is a result of particle selection and preferential ingestion and the rapid elimination of microplastics in the species. And certainly humans are ingesting and inhaling microplastics. However, the health risks are really not known, specifically when you're talking about the number that we are ingesting and inhaling on a monthly or yearly basis. Okay, and with that, I'm going to just acknowledge some people that have helped me with these studies, some industry members that have also been involved in our work, and funding agencies that have supported the studies that I've shown you today. So with that, I'm going to stop sharing my screen and turn it back over to Dennis.
Thanks, Evan. Fascinating. Loved the endoscopic video. That was so cool.
Thanks.
So if anybody has any questions, you're welcome to unmute yourself and ask.
And I see some. Are there some in the chat? Let's see.
No, they're all links that
Nope.
I put in.
No, they're all links. Okay.
Questions, anybody?
So, Evan, what do you see as the extension of the work that you're doing, and who else locally around Long Island Sound is participating in these studies?
Yeah, so the answer to the first question is what we're trying to do is identify a good bioindicator of microplastics in the environment. So if you look in the literature, many people have expressed their opinion that bivalves are a good bioindicator of microplastics in the environment because bivalves are relatively easy to collect, right? They're out in the environment all the time. They do process large volumes of water, and they're found worldwide. Some bivalve can be found pretty much anywhere worldwide. However, our work and our arguments are that no, because of particle selection, they are not a good bioindicator, as I've shown you. So if you go out in the environment and pick up a bivalve, that is only showing you a small snapshot of what's in the environment because of particle selection and rapid elimination. So the large part of what we've been doing recently is trying to find other organisms that are easy to collect, are suspension feeders, and that are non-select suspension feeders. So we've been working with tunicates, for example. Tunicates can be found, sea squirts can be found all over the world. They are, for the large part, non-select particle feeders. And so we're looking at whether they can be a good bioindicator. Also, we're looking at slipper snails. That's a snail, but it's a suspension feeding snail. Again, some species of slipper snail can be found pretty much worldwide. And they are also pretty much non-select particle feeders. So we're doing the same types of experiments I just showed you with bivalves on these other organisms to see if we can determine a good bioindicator. The other thing we're trying to do is we're using, I only showed you a small subset of the data because we only used a couple different particles, but we've expanded those studies with different particle shapes, sizes, and polymer types. And we're trying to develop a model that will be able to predict which types of microplastics in the environment the bivalves actually ingest and which ones they reject. Because if we can determine that, then we can say, for bivalves at least, we can say, well, you know, this certain type of microplastic is one that they always ingest. So that's one we really need to take a hard look at and see if we can eliminate from the plastic stream, right?
Yep.
So that's the answer to the first part. The second part is who else is doing this work is a couple other people in Connecticut that are doing microplastic work. Certainly in the region, there are people at Woods Hole that are doing. I collaborate with them at URI up in Massachusetts as well. Within our department here, Heidi Dearson just got a grant to look at whether microplastic concentrations can be determined using satellite imagery. So she works on satellites and satellite imagery. And you've probably seen, many people have seen those, figures, the views of satellite imagery where they can determine the concentration of phytoplankton. You can see, a warm area of the water and cold areas of the water and stuff. And so there may be a way that you could fine tune satellite imagery to help us determine where the microplastics are and what their concentration is. So that's very interesting. She just started that work. It's an interesting sort of take on it.
What about jellyfish?
As far as uptake, you mean?
Right.
Uptake of microplastics by jellyfish. So there's a person at Roger Williams University that has done a little bit of work on the uptake of microplastics by jellyfish. And, unsurprisingly, if you give them a high concentration of microplastics, some end up in the gut. I'm not so sure we know what the concentration would be if you give them environmentally relevant concentrations of microplastics, just because there's so few microplastics. And they are passive suspension feeders, of course, not active suspension feeders. So they process a lower quantity of water per unit time than something like a bivalve or a tunicate or something like that, because they're passive.
Right.
So related to what you're just talking about, it's a question, what does suspension feeding mean?
So that's the same as filter feeding. You can think there's actually a difference, but the difference is technical, and so it's the same as filter feeding. Yeah, you can just think of it as filter feeding.
Amy asks, is there any hope to reduce microplastics already in the water, and how would we do that?
Yeah, I'm not so sure there is. Not in the natural environment. I think the key is to try to reduce the input of macroplastics, so there's a lot fewer that are breaking down into microplastics. Now, after saying that, I have a new project with the engineers at UConn in which we're looking at the feasibility of using freshwater bivalves to remove microplastics from treated sewage effluent, so that the sewage effluent, after it was treated to some degree, would pass over beds of freshwater mussels to remove the microplastics and reduce the amount of microplastics that are entering the waterways. So if you have something confined like that, like a sewage treatment plant, I think there's a good chance that you could remove more of the microplastics than what is being removed now.
Where
But in the natural environment, I don't know.
Now, the sewage treatment plant that you're doing this, Riz, is the one at Avery Point, affiliated with Avery Point?
No, it's the one up at Stores.
Okay,
So Stores has its own sewage treatment plant.
right,
Yeah, so we have access to that. We're also looking at the bacteria that can be found in the gut and in the feces of bivalves after they feed on microplastics, to see if we can identify bacteria that could be used in a biofermenter to break down microplastics or plastic material in general. So that's what the engineers.
Anybody else have a question?
excellent presentation, Evan.
Thank you,
Okay,
Evan.
thank you.
For doing this
Sure.
for us.